Inside a mountain in Romania, there is a glacier that has never seen daylight. It sits underground, in total darkness, inside a cave so large it could swallow 40 Olympic swimming pools. It has been frozen continuously for roughly 13,000 years. And in early 2026, scientists drilled straight down into it, pulled out a single sliver of ice from 5,000 years deep, and woke something up that should not exist. What they found wasn't a virus or a fossil or a curiosity for a museum case. It was a living bacterium, fully alive. And when researchers tested it against 10 of the antibiotics modern medicine depends on most, the 5,000-year-old microbe simply shrugged them off, one after another, as if it had been waiting for this exact fight the entire time. This is the story of how something older than written language turned out to already know how to survive medicines that wouldn't be invented for thousands of years. And what that terrifying coincidence is actually telling scientists about the future of infectious disease. Let's start with the place itself, because the location matters as much as the discovery. The cave is called Scărișoara, tucked into Romania's Apuseni Mountains. Inside it sits a body of perennial ice roughly the size of an apartment building, a frozen archive that has been quietly accumulating layer after layer for thousands of years, completely undisturbed by sunlight, weather, or human activity. To study it, researchers drilled a 25-meter ice core straight down through the cave's Great Hall, extracting a continuous timeline of frozen history stretching back roughly 13,000 years. Essentially a tree ring record, except made of ice instead of wood, with each layer representing a slice of time sealed away the moment it froze. Deep inside that core, at a layer dated to approximately 5,335 years old, researchers led by microbiologist Cristina Purcarea at the Institute of Biology Bucharest found something alive. A bacterium belonging to a genus called Psychrobacter. Organisms specifically adapted to thrive in extreme cold. The kind of microbe you'd expect to find surviving in glaciers, polar soil, or the deepest, coldest parts of the ocean. This particular strain got a name, Psychrobacter SC65A.3. To be extracted safely, every step had to preserve the sample exactly as it was found. The ice was kept frozen during transport, handled with extreme care to avoid modern contamination. Then thawed only under sterile laboratory conditions, designed to ensure that whatever grew afterward had genuinely survived inside that ice for 5 millennia, rather than sneaking in from the modern world. Once researchers had the living strain in hand, they did the obvious thing: they tried to kill it. Specifically, they exposed it to 28 different antibiotics spanning 10 distinct drug classes, essentially the modern medical arsenal used against serious bacterial infections. The results should unsettle you a little. The ancient bacterium showed resistance to 10 of those antibiotics, spanning 8 different drug classes. Not mild resistance. Functional, working resistance to medications including rifampicin, vancomycin, and ciprofloxacin. Antibiotics doctors currently rely on to treat tuberculosis, severe colitis, and urinary tract infections in actual hospitals, on actual patients today. When researchers sequenced its genome, they found the bacterium was carrying more than 100 genes linked to antibiotic resistance mechanisms. Here's the detail that should stop you cold. This bacterium had been frozen for roughly 5,000 years. Penicillin, the first antibiotic ever discovered, wasn't identified until 1928. This organism had never once, in its entire evolutionary history, been exposed to a pharmaceutical antibiotic, and yet it was already carrying the genetic toolkit to defeat them. How is that possible? The answer reframes something most people get backward about antibiotic resistance. Most public conversation treats resistance like something humans accidentally invented through overprescribing antibiotics, as if bacteria were innocent until modern medicine provoked them. But resistance genes didn't appear the moment humans started manufacturing penicillin in a lab. They are ancient survival tools present in microbial populations for millions of years, originally evolved for reasons that have nothing to do with hospitals or prescriptions at all. In the wild, bacteria are constantly at war with each other and with fungi, which naturally produce antimicrobial compounds as chemical weapons in their own competition for resources. A bacterium living in soil, ice, or rock has been fighting this microscopic chemical war since long before humans existed, let alone before humans figured out how to bottle and prescribe the same compounds. The genes that let SC65A.3 survive vancomycin weren't built to defeat human medicine. They were built over unimaginable stretches of evolutionary time to survive a world already saturated with natural antimicrobial weapons that simply happen to share mechanisms with our modern drugs, what humanity calls the antibiotic era. Roughly the last 100 years is, from a bacterium's perspective, just one more recent skirmish in a war that has been running for millions of years, fought largely out of sight, in soil and ice and rock, long before anyone in a lab coat got involved. This is precisely why discoveries like SC65A.3 matter far beyond scientific curiosity. Environments like ancient ice, what researchers call cryo-environments, function as enormous, largely unexplored genetic archives. Frozen ground, glacial ice, permafrost, these aren't simply frozen wastelands. They're vast natural databases of resistance genes that have been quietly evolving and accumulating for thousands, sometimes millions, of years, almost entirely unstudied until recently, because nobody had a strong reason to drill into them and look. Which raises an uncomfortable but important question for modern medicine. As global temperatures rise and previously stable ice and permafrost begin to thaw at an accelerating rate, what else is being released that has been quietly evolving defenses against threats most modern bacteria haven't even encountered yet? But, and this is the part that turns the story from a warning into something closer to an opportunity. Purcarea's team didn't stop at documenting resistance. They also found that SC65A.3 actively inhibits the growth of several modern antibiotic-resistant superbugs, and that it carries enzymatic activity with real biotechnological potential. In other words, the very same ancient survival mechanisms that make this bacterium nearly impossible to kill with current antibiotics might also contain entirely new biochemical tools scientists could study and adapt into the next generation of treatments. Medicines designed for a world where today are steadily losing antibiotics are sent their effectiveness. It's a strange kind of irony. A microbe that has spent 5,000 years quietly proving that nature invented antibiotic resistance, long before humans invented antibiotics, might also help us build the next class of drugs capable of getting around it. So picture that ice core again, 25 meters of solid frozen time, pulled up out of total darkness, layer by layer, each one a sealed capsule from a year nobody alive was there to witness. Somewhere around layer after layer down, past 5,000 years of sealed silence, something was simply waiting, not extinct, not fossilized, alive the entire time, holding a genetic memory of battles fought long before the first antibiotic was ever discovered. A reminder that the war between microbes and medicine didn't start with us.